There is an animal sitting perfectly still on a branch right now, watching two completely separate worlds simultaneously, without moving a single muscle in its neck. Each of its eyes is doing something different. One is scanning the canopy above, the other is watching the forest floor below, and the information from both arrives in its brain at the same time, processed in a way that no other vertebrate on Earth has quite replicated. The chameleon has been quietly extraordinary for a very long time, and most of what makes it extraordinary happens slowly, almost invisibly, in the filtered green light of forests that are themselves half asleep. Tonight, we will follow it from the first pale light of morning through its long, measured day, into the color it wears, the tongue it fires, and the strange, ancient machinery of a body built entirely around patience. So, let the room settle around you now. Let your eyes grow heavy, and drift with me into the dappled stillness where the chameleon has already been waiting, watching two directions at once, perfectly unhurried. The chameleon's eyes are unlike those of almost any other vertebrate alive today. Each one sits in a small, cone-shaped turret that protrudes from the side of the head, covered entirely by a scaled, fused eyelid with only a small circular opening at the center through which the pupil is visible. These turrets are capable of rotating independently through almost 180 degrees in the horizontal plane and roughly 90 degrees in the vertical, giving each eye a field of view that sweeps across most of the hemisphere on its side of the body. When the two eyes move independently, the chameleon can cover virtually the entire sphere of space around itself simultaneously, with no blind spots except directly behind the tail. No neck movement is required, no repositioning of the body. The world arrives continuously and completely, from every direction at once. This independent eye movement is called monocular vision, and in the chameleon it reaches an extreme rarely seen elsewhere in the vertebrate world. Most animals with laterally placed eyes, which includes most non-predatory species, have some degree of independent eye movement. But the range and independence of movement in chameleons is exceptional. The two eyes are controlled by independent sets of extraocular muscles, and the neural wiring that coordinates them allows each eye to pursue its own target, track its own subject of interest, without any obligatory coupling to what the other eye is doing. A chameleon resting in a bush can simultaneously monitor a distant branch for the approach of a rival with one eye and track a slow-moving beetle on the leaf below it with the other, processing both streams of visual information in parallel. The visual processing that underlies this independent monitoring is itself remarkable. In most animals, including humans, the two eyes' inputs are integrated into a single fused image in the brain, requiring both eyes to point at approximately the same region of space for the system to function well. In chameleons, the two visual streams remain largely separate in the brain during the scanning phase, each processed in its own hemisphere without obligatory fusion. But when the chameleon detects prey with one eye and switches to a targeting mode, the two eyes converge toward the same point, and the brain shifts from its parallel scanning mode to a binocular depth assessment mode. This convergence is the behavioral moment that researchers who study chameleon hunting behavior call fixation, and it marks the transition from the chameleon's wide-area surveillance mode into something much more precise and intentional. The optics of the chameleon eye have been studied in considerable detail, and they reveal a system optimized for high resolution across a large field, rather than the high sensitivity, low light design found in nocturnal animals. The lens of the chameleon eye is relatively small in relation to the eye's overall size, and the retina is densely packed with cone cells, the photoreceptors responsible for color vision and fine spatial detail. The fovea, the small central region of highest acuity, is unusually well developed in chameleons compared to other lizards. This investment in resolution over sensitivity reflects the chameleon's primarily diurnal lifestyle and its reliance on detecting the subtle movements of insects against visually complex backgrounds of leaves, bark, and filtered light. One of the more technically interesting features of chameleon vision is how the animals appear to cope with the potential for visual confusion when one eye is tracking a moving prey item while the other is scanning for predators. The neural architecture that allows this seems to involve a degree of attentional gating, in which the relevance and urgency of each eye's input is weighted dynamically, with high salience targets, a nearby predator, or a clearly stationary prey item able to redirect the animal's motor response, even when the other eye's stream is also active. Researchers studying chameleon gaze behavior have observed that prey detection in one eye reliably triggers a reorientation of both eyes toward the target, even if the other eye was actively tracking something else moments before. The system coordinates from separation to unity when the moment demands it. There is a quality to the chameleon's visual experience that we can only approximate from the outside, but it must involve a kind of spatial awareness radically different from anything familiar to us. Imagine standing in a forest and being able to see everything behind you, to your left, to your right, and in front, all simultaneously, with each side of your visual field in sharp focus and under active, conscious surveillance. The chameleon lives this as its ordinary experience of the world. The forest is not something it must turn its head to scan. It is something it simply inhabits, present to it in all directions at once, a continuous panoramic clarity that requires no effort to maintain. This constant all-surrounding vision is perhaps part of what makes the chameleon so extraordinarily patient. It already knows what is there. Color change in chameleons is one of the most visually dramatic phenomena in the animal kingdom, and also one of the most widely misunderstood. The popular conception that chameleons change color to match their surroundings in the way a pixel adjusts to blend into a background image is not quite right. What chameleons actually do is both more complex and more interesting. Their color changes are primarily communicative and physiological, rather than purely camouflage-driven, and the mechanism that produces them involves a layer of physics and biology that was not fully understood until surprisingly recently, a discovery that required electron microscopy and crystallography to properly see. For most of the 20th century, scientists understood that the color cells of chameleon skin, called chromatophores, contained pigments and reflective granules that could be repositioned or concentrated to alter skin color. This model, essentially the same as the one used to describe color change in cuttlefish and other cephalopods, worked reasonably well for the darker, browner tones that some chameleons produce under stress or cooling. But it struggled to fully explain the brilliant blues, turquoises, and whites that male chameleons display during social interactions. In 2015, a team led by Michel Milinkovic at the University of Geneva published a paper that explained the mechanism behind these iridescent colors. And it turned out to involve something no one had quite anticipated: tunable photonic crystals. Beneath the outer layer of pigment-containing chromatophores in chameleon skin lies a second layer of cells called iridophores, each containing a three-dimensional lattice of guanine nanocrystals arranged in a regular geometric pattern. When white light strikes this lattice, it interferes constructively at specific wavelengths determined by the spacing of the crystals, reflecting those wavelengths back to the observer while other wavelengths cancel. This is structural color, the same physical principle that makes a soap bubble iridescent or a peacock feather brilliant, produced not by pigment, but by the geometry of a physical structure. The remarkable thing about the chameleon's iridophores is that the spacing of the crystal lattice is not fixed. When the cell relaxes or contracts in response to hormonal signals, the crystal spacing changes, shifting the wavelength of reflected light across the visible spectrum from red through orange and yellow to green and blue. The speed at which a chameleon can shift its structural color is faster than is often appreciated. Under experimental conditions, with appropriate hormonal stimulation, color shifts can begin within seconds, and the full transition from one color state to another can occur within a minute or two. In natural behavioral contexts, dominant male veiled chameleons, displaying to rivals, can cycle through multiple color patterns over the course of a short aggressive interaction. Researchers who have quantified the relationship between color pattern and social outcome in these interactions have found that the speed and amplitude of the color shift, how quickly and dramatically the male changes, is a predictor of the interaction's outcome, with faster, brighter shifting correlating with dominance. The color change is not decorative. It is a signal whose dynamics convey information about the signaler's state and condition. Female chameleons also change color, though generally with less dramatic amplitude than males. A significant color change in females occurs during pregnancy, when gravid females of many species develop vivid patterns of black, orange, and blue that serve as a signal to approaching males that the female is not available for mating. This gravid coloration is under hormonal control and appears involuntarily. The female does not choose to display it. It is simply the color her skin produces when the hormonal state of pregnancy activates the iridophore lattice in a particular configuration. In some species, bun-receptive females that are not pregnant can produce similar patterns voluntarily in response to male harassment, suggesting a degree of volitional control over the system in social contexts, even when the pattern's original function was physiological. The chameleon's skin is, in this sense, something closer to a screen than a surface. It receives signals from the nervous and endocrine systems, translates those signals into optical output through the physics of its crystal lattice, and projects a message outward to any observer with the right visual system to receive it. That message can say many things: warmth-seeking, territorial, receptive, aggressive, stressed, or gravid. It can carry information about the animal's age, condition, and social status, and it updates continuously, shifting in real time with the animal's internal state. The skin thinks in light, and any chameleon looking at another is reading, in the most literal optical sense, the thoughts broadcast across its surface. The chameleon's tongue is one of the most extraordinary ballistic structures in the vertebrate world. It can extend from the mouth to a distance equal to or greater than the animal's body length in a fraction of a second, strike a prey item with a sticky, muscular tip, and retract back into the mouth with the prey secured, all before a human observer watching in real time would have seen anything more than a blur. The process has been measured with high-speed cameras at speeds exceeding 26 body lengths per second, with peak accelerations reaching more than 41 g, a force that would render a human unconscious. The physics required to produce this performance involve a biological mechanism that engineers studying fast-launching systems have found genuinely instructive. The tongue launch mechanism is based on elastic energy storage rather than direct muscular contraction. At the core of the tongue is a rod-shaped accelerator muscle surrounding a central hyoid bone extension. When the chameleon prepares to strike, the retractor muscles that normally hold the tongue in the mouth contract and stretch a series of collagenous sheets surrounding the accelerator muscle, loading them with elastic potential energy like a compressed spring. When the trigger signal arrives, this stored energy is released all at once, driving the accelerator muscle and the tongue's outer sleeve forward along the hyoid rod in a single explosive discharge. The muscular energy required to load the spring and the elastic energy released in the strike are decoupled in time, which is what allows the tongue to achieve accelerations that no direct muscular contraction could produce on the same timescale. The sticky tip of the tongue, called the tongue pad, is a specialized mucus structure that adheres to insect prey through a combination of wet adhesion and suction. The mucus is unusually viscous, estimated to be roughly 400 times more viscous than human saliva, which allows it to maintain contact with the prey during the brief moment of impact and the subsequent retraction. Studies using high-speed footage and force measurements have found that the adhesive force at the tip scales with prey mass in a way that suggests the mechanism is not purely passive adhesion, but involves some active suction component generated by the musculature of the tongue pad itself. The chameleon does not simply stick its tongue to a cricket and pull. It grabs in a soft and almost hydraulic sense, and the grip is extraordinarily reliable across a wide range of prey sizes and surface textures. The accuracy of the tongue strike requires the chameleon to have precise distance information before launching, which is why the binocular convergence phase that precedes a strike is so behaviorally important. Researchers studying the visual control of the strike have found that chameleons use stereoscopic depth perception, the same principle humans use for near-space depth assessment, during the targeting phase, with the degree of convergence of the two eyes providing a direct neural signal about the angular distance to the prey. This information is fed directly into the motor programs that control tongue projection distance, essentially calibrating the strike length to the measured distance. The whole system from visual input to tongue launch is so tightly integrated that the entire targeting and launching sequence, from the moment the eyes converge to the moment the tongue contacts prey, can occur in under a second. Temperature has a significant effect on tongue performance. Like all physiological processes in ectothermic animals, the biochemical reactions underlying muscle contraction and elastic recoil in the tongue are temperature dependent. At lower temperatures, the tongue launches more slowly and with less force, potentially reducing both range and accuracy. This is one of the reasons chameleons engage in extended basking behavior at the beginning of each day before beginning to hunt. A chameleon at full thermal operating temperature can project its tongue accurately and powerfully. A cold chameleon is a compromised hunter. Research measuring tongue performance across a temperature range found that the tongue strike of a cold chameleon can be dramatically impaired, with some individuals failing entirely to extend the tongue effectively at temperatures significantly below their thermal optimum. There is something almost meditative about watching a chameleon hunt, even knowing what the tongue can do. The animal sits motionless for long periods, tracking the movements of potential prey with its eyes, while its body remains completely still. The stillness is functional, both to avoid alerting the prey and to maintain the accuracy of the visual targeting system, which relies on a stable platform. When the moment comes, there is a brief, almost imperceptible shift in the position of the eyes, a final binocular lock onto the target, and then the tongue is already gone and back before the movement has fully registered. The prey vanishes. The chameleon resumes its stillness. The entire event is over in less time than it takes to blink, and yet it represents the culmination of minutes or hours of patient, precise, thermally optimized preparation. The strike is brief. Everything around it is not. The chameleon's feet are built for a very specific kind of world. Narrow, rounded branches in a three-dimensional arboreal environment where slipping has immediate and serious consequences. Each foot is divided into two opposed groups of toes, with 2 toes on one side and 3 on the other, in a pattern called zygodactyly, the same toe arrangement found in parrots and woodpeckers. These opposed toe groups work together as a gripping tong, closing around a branch from both sides simultaneously and generating a secure hold without requiring the animal to actively squeeze. The grip is largely passive once established, with the toes locking into position through the geometry of their articulation, rather than requiring continuous muscular effort to maintain. A sleeping or basking chameleon can hang securely from a perch without burning energy, holding on. The chameleon's gait on branches is one of the more distinctive locomotor patterns in the reptile world. The animal moves with a characteristic rocking motion, shifting its weight slowly from one side to the other with each step, advancing one leg at a time in a deliberate sequence. This rocking is not random. It is thought to serve a specific visual function. By introducing controlled motion to its own visual field, the chameleon may be using the resulting visual flow patterns to extract depth and distance information from the environment in a way that a completely stationary observer cannot. The rocking essentially creates a form of motion parallax, the apparent shift in the relative positions of near and far objects as the viewpoint moves, which provides three-dimensional spatial information about the branch structure ahead. The tail of many chameleon species is prehensile, functioning as a fifth limb that can coil around a branch and anchor the animal independently of its feet. This prehensile capacity involves a muscular and skeletal tail structure quite different from the non-gripping tails of most other lizards. The tail can curl into a tight spiral for storage when not in use, and can extend and coil around a branch independently in response to instability events like sudden gusts of wind or unexpected contact. In some species, the tail is proportionally longer and more muscular than in others, and correspondingly more central to the animal's locomotor security in complex vegetation. In the moment when a chameleon extends its tongue at a prey item, the tail anchors to the perch, ensuring that the recoil from the tongue launch does not destabilize the body. Chameleon locomotion on uneven or unpredictable branch surfaces involves continuous sensory feedback from the feet and tail, updating the animal's motor programs in real time. Pressure receptors in the toe pads and proprioceptive sensors throughout the limbs provide constant information about the quality of the current grip and the distribution of body weight across the support points. This feedback allows the animal to detect early signs of grip failure and respond with corrective adjustments before a false slip occurs. Researchers studying chameleon locomotion have found that the animals vary their grip force and limb positioning in response to experimentally manipulated surface properties in ways that demonstrate active sensory motor regulation rather than stereotyped mechanical movement. The slowness of chameleon movement, so distinctive that it has become one of the animal's defining characteristics, is not simply laziness or low metabolic tempo. It reflects a carefully calibrated strategy for moving through an environment where detection by predators and prey alike depends critically on motion. Many of the visual systems that chameleons need to avoid, including those of predatory birds, are highly sensitive to motion against a static background. By moving so slowly that the motion falls below the detection threshold of these systems, the chameleon effectively renders itself invisible to predators, even in broad daylight in exposed positions. The same principle applies in reverse to the chameleon's own hunting. By remaining merely stationary while tracking prey, it avoids triggering the escape responses that most insects initiate in response to approaching movement. The overall locomotor strategy of the chameleon is one of minimum movement, maximum grip security, and continuous sensory updating, executed at a pace that the surrounding environment rarely perceives as motion at all. This is not the locomotion of an animal constrained by poor design. It is the locomotion of an animal that has optimized its movement strategy to match the specific demands of living in slow, filtered light among dense vegetation where patience is more productive than speed. Every step is placed with intention. Every grip is tested before weight is committed. Every pause is an opportunity for the eyes to gather information and for the tongue to prepare. The chameleon moves the way the forest itself seems to move, in increments so slow they are almost imperceptible, and yet always precisely arriving. The chameleon is ectothermic, meaning it cannot generate body heat internally the way mammals and birds do. Its body temperature is determined almost entirely by its thermal environment, and the management of that temperature is one of the central organizing tasks of its daily life. This is not a deficiency, it is a different strategy, one that trades the high energetic cost of endothermy for a metabolic rate low enough that a chameleon requires far less food than an equivalently sized mammal to sustain itself. But it does mean that the chameleon's world is divided, in a practical sense, into thermal zones of varying quality, and that the animal must navigate between them continuously to remain functional. The morning routine of a basking chameleon reveals just how precisely this thermal management operates. Before sunrise, a chameleon typically rests in a position oriented to intercept the first direct sunlight of the day, often on an exposed perch on the outer surface of vegetation where the morning sun will strike it. As light arrives, the chameleon's skin darkens. This darkening is caused by the dispersion of melanin-containing pigment granules in the superficial chromatophore layer, the same cells involved in color change, but operating here in the infrared management mode rather than the signaling mode. Darker skin absorbs solar radiation more efficiently than lighter skin, and the thermal benefit of this pigment shift can be measured directly. Dark-skinned basking chameleons heat up significantly faster than they would if their skin remained at its normal, lighter active phase color. The target body temperature for most chameleon species falls in a fairly narrow band, typically somewhere between 28 and 35 degrees Celsius depending on the species, and the animal's behavior is organized around reaching and maintaining this temperature during the active period of the day. A chameleon that overheats moves into shade. One that cools below the optimal range moves back towards sunlight or onto a surface that is warmer from solar radiation. Throughout the day, this behavioral thermoregulation continues as a low-level background activity, shaping the animal's position and activity level in ways that may not be obvious to an observer watching its apparently random movements. What looks like a chameleon sitting on one branch and then relocating to another for no apparent reason is often, in fact, a precise thermal management decision. Temperature regulation interacts with digestion in ways that are particularly significant for chameleons. Like all ectotherms, chameleons digest their prey most efficiently at or near their optimal body temperature. A chameleon that has eaten but is cold cannot process its meal effectively, and the food sitting undigested in its stomach becomes a metabolic liability rather than an asset. This coupling between temperature and digestive efficiency means that access to appropriate thermal environments is not simply a matter of comfort, but a physiological necessity following feeding. Some researchers have noted that chameleons appear to preferentially seek higher body temperatures immediately following a meal, essentially using behavioral thermoregulation to accelerate their digestion and the conversion of prey into usable energy. Nighttime thermal management presents a different set of challenges. As temperatures drop after sunset, chameleons move toward the interior of vegetation, selecting roosting positions that minimize radiative heat loss, or in some species, descending to lower, warmer microclimates near the ground. At the roosting temperature, the animal's metabolic rate drops significantly, reducing both energy consumption and the requirement for oxygen. The respiratory rate slows, heart rate decreases. All non-essential physiological processes throttle back. The chameleon enters a state that is not quite sleep in the mammalian sense, but is a sustained low-activity rest that allows the body to conserve the thermal and metabolic resources it will need to perform the warming-up sequence again at dawn. There is a broader ecological architecture to the chameleon's thermal world that becomes visible when we zoom out from the individual animal to the structure of the habitat it inhabits. The forest is not thermally uniform. It contains patches of sunlight and deep shade, surfaces warmed by direct radiation, and surfaces cooled by evaporation. Thermal gradients that shift through the day as the sun moves and the canopy casts changing shadows. The chameleon navigates this thermal landscape with a sophistication that reflects a continuous, ongoing perception of its thermal environment. In a sense, the forest looks different to a chameleon, not just because of the independent panoramic vision of its eyes, but because every surface and every patch of light carries thermal information that is continuously integrated into the animal's behavioral decisions. The world, seen through this lens, is a thermal map as much as a visual one. Most animals that study chameleons with their eyes are missing something important. The visual system of a chameleon includes receptors sensitive to ultraviolet light, a range of wavelengths that is invisible to human vision, but that occupies a significant portion of the electromagnetic spectrum above the violet end of what we can see. Many of the color signals that chameleons produce involve ultraviolet components, particularly in the structural colors generated by the iridophore layer. When researchers photograph chameleon displays using cameras that capture UV wavelengths, they frequently see patterns in the UV that have no visible light equivalent, patterns that are invisible to a human observer watching the same interaction, but clearly present in the recorded data. The significance of UV signaling in chameleons has been explored through a series of elegant experiments in which researchers altered the UV component of illumination in enclosures containing live chameleons and observed the behavioral effects. Chameleons deprived of UV light, even when ambient visible light levels were unchanged, showed altered social behavior in ways consistent with the loss of important information from the visual signal. Male-male interactions in particular appeared affected, with the usual assessment rituals of color display disrupted in UV-deprived conditions. These experiments suggest that the UV component of the chameleon's color display carries information that is functionally significant for the receiver, not simply an incidental byproduct of the structural color mechanism. The ability to see UV is linked to the presence of a specific photoreceptor class in the eye, containing a visual pigment with peak sensitivity in the ultraviolet range. This receptor class, known as a UV or VS cone, is present in the eyes of most lizards and many other vertebrates, including birds, but is absent in the lineage leading to mammals, where it was apparently lost early in the evolutionary history of the group. Humans and other primates lack UV vision, partly as a result of this ancient loss, and we see a spectrally impoverished version of the chromatic world compared to most other vertebrates. The chameleon, with a full complement of 4 cone types, including a UV-sensitive class, occupies a richer color world than we can directly experience, and the signals it sends and receives carry information in frequencies we can only detect with instruments. The structural organization of chameleon color displays has been quantified by researchers using spectrophotometry and visual modeling to reconstruct what the signals look like from the perspective of the receiver's visual system, rather than a human observer's. These models incorporate the known spectral sensitivities of the chameleon's 4 cone types and the ambient light spectrum in the animal's habitat to predict which color differences would be most salient and discriminable. The results often differ substantially from what a human would intuitively describe as the most conspicuous elements of the display. A pattern that looks to a human like a subtle blue tinge may be an intensely UV reflective signal that dominates the chameleon receiver's color perception. What we see when we watch a chameleon display is a translation, and something is always lost in translation. Chameleons that live in different light environments have been found to produce color signals whose spectral properties are adapted to transmission in their specific habitat. Species living in closed canopy forest, where UV is largely filtered by the leaf canopy, tend to rely more on longer wavelength structural colors that transmit effectively through the predominantly green-shifted ambient light. Species living in more open, UV-rich environments tend to show more elaborate UV signal components. This habitat-correlated signal evolution reflects the same principle that shapes bird plumage and flower color in relation to pollinator vision. Signals evolve to exploit the most reliable and detectable channels available in the ambient light environment where communication occurs. The chameleon's color vocabulary, in other words, is not arbitrary. It is shaped by the physics of the forest it lives in. There is something quietly humbling about recognizing that the chameleon is, in part, having conversations we cannot hear in frequencies of light we cannot see, on surfaces that transform their appearance in real time in response to internal states we can only infer. Our perception of the chameleon display is incomplete, necessarily, and we have only recently developed the tools to begin filling in the parts we were missing. This is true more broadly of the natural world, of course, in that almost every species perceives its environment through sensory channels that differ from our own, and almost every interaction in nature carries information in frequencies and modalities that exceed the human sensory envelope. The chameleon is simply a particularly vivid reminder of this truth, dressed in light we cannot fully see, speaking in colors that extend beyond the edge of our world. Watching a chameleon at night is a different experience from watching one during the day. Something changes, visibly and dramatically, in the hours after sunset. The color that the animal wore during its active period, whether the muted greens and browns of its resting camouflage or the brighter hues of a social interaction, fades and reorganizes. The skin often lightens and sometimes develops a distinctive pale pattern, bands or patches that were not present during the day, arranged in ways that vary between species and individuals, but that are remarkably consistent from night to night in the same animal. This nocturnal color pattern is not a display and not camouflage in any obvious sense. It appears to be a physiological state rather than a social signal, and its specific function and mechanism have been subjects of active scientific interest. The nighttime color pattern is thought to be related to the reduced neural and hormonal control of the chromatophore and iridophore system during sleep. During active periods, the chameleon's color is under continuous modulation by the nervous system and by circulating hormones, including melatonin and various stress-related compounds. At night, as the animal enters its rest state and hormonal levels shift toward the sleep-associated profile, the active control of the skin cell system may relax into a default configuration that is partly set by the passive optical properties of the crystal lattice in a low excitation state. The resulting pattern is, in a sense, what the skin looks like when nothing is actively changing it. A resting face, worn during the hours when the forest is dark and the audience of conspecifics and predators is mostly inactive. There is an interesting line of research suggesting that chameleon sleep color patterns may serve a thermoregulatory function during cool nights, with lighter coloration at night reflecting more infrared radiation than the dark basking color would, helping the animal avoid excessive radiative cooling to a cold night sky. The physics of this is straightforward. A lighter, more reflective surface loses less heat by radiation than a darker, more absorptive one. If the nighttime pattern is consistently lighter in individuals of populations that experience cold nights, this would support the thermoregulatory hypothesis. Some comparative data across chameleon species from different thermal environments does suggest this kind of correlation, though the evidence is not yet comprehensive enough to be definitive. Chameleon sleep itself, as a behavioral state, has been relatively little studied compared to the rich literature on chameleon color change and foraging. What is known is that chameleons select specific roosting sites at dusk, typically returning to the same perch or the same small area of vegetation, night after night. The selection criteria for roosting sites appear to include thermal properties, structural security against predators, and possibly proximity to the previous day's foraging area. Once settled, the animal assumes a characteristic resting posture, with its eyes closed and its body pressed gently against the branch, tail curled, and limbs positioned symmetrically. The regularity of this routine across nights suggests a learned spatial memory for suitable roosting locations, rather than a fresh search each evening. The eyes of a sleeping chameleon provide one of the more charming visual details of their nighttime biology. The distinctive scaled eyelids, each covering the eye as a single fused cone-shaped structure, with only the small central pupil opening remaining, close the pupil opening completely during sleep, giving the resting chameleon's eye the appearance of a smooth, seamless dome rather than the rotating turret visible during active periods. Some species show brief eye movements detectable through the eyelid during sleep, raising the possibility that chameleons experience something functionally analogous to the rapid eye movement sleep found in birds and mammals. Though the neural basis and significance of any such movements in a brain as differently organized as a chameleon's would need careful, independent analysis. The transition from sleep back into wakefulness at dawn involves a gradual process of thermal and behavioral activation that is worth appreciating in detail, because it reveals how deeply the chameleon's physiology is organized around light and temperature together. As the first light reaches the sleeping animal, the skin responds before the animal is fully awake, darkening slightly as melatonin levels drop and pigment granules begin dispersing in response to the changing hormonal state. As body temperature rises with incoming solar radiation, metabolic rate increases, neural activity picks up, and eventually the eyes open, each one independently scanning the brightening forest before the animal has moved a limb or changed its posture. Wakefulness arrives from the outside in, written in light on the skin before it fully reaches the brain. Chameleons belong to a family called Chameleonidae, and their evolutionary history within the lizard lineage has been a subject of considerable paleontological and molecular interest. For a long time, their fossil record was frustratingly sparse, because chameleons' small, lightweight bones do not preserve well, and many species have lived in forested environments where fossilization conditions are poor. But a combination of molecular clock analyses and the occasional exceptional fossil find has gradually assembled a picture of the group's deep history, one that is both older and more geographically complicated than the popular image of chameleons as an African group might suggest. Molecular clock studies, which use the rate of genetic mutation as a proxy for elapsed time, place the origin of modern chameleons at somewhere around 80 to 90 million years ago, in the late Cretaceous period. This means that chameleons were already living in forests that also contain the last non-avian dinosaurs, exploiting arboreal niches and ecosystems that no longer exist. What those original Cretaceous chameleons looked like, and how their distinctive features, the compressed body, the turret eyes, the prehensile tail, and the ballistic tongue, were distributed across their early evolutionary radiation, is only partially known from fossils. But the molecular evidence clearly places the lineage as an ancient one, already distinct from its lizard relatives before the extinction event that ended the Mesozoic. The early geographic history of chameleons is one of the more intriguing puzzles in lizard biogeography. The greatest diversity of chameleon species today is found on Madagascar, which is home to more than half of all known chameleon species, and contains many of the most morphologically extreme forms. The African mainland has a rich chameleon fauna as well, including the genus Chameleo and the bizarre leafy Rampholion group. A smaller number of species extend into the Middle East, the Indian subcontinent, and southern Europe. This distribution pattern, with a major center of diversity on an island that has been isolated from mainland Africa for tens of millions of years, has been interpreted as evidence that the chameleon lineage originated in Gondwana and dispersed across the ancient southern landmass before the breakup that created the Indian Ocean and the modern arrangement of continents. Fossils of early chameleons discovered in Europe, including specimens from Miocene deposits in Germany and other sites, reveal that chameleons had a wider geographic distribution in the geological past than they do today, extending across what is now Europe during periods of warmer climate. The contraction of chameleon range to its current tropical and subtropical distribution appears to be a relatively recent phenomenon on geological timescales, associated with the progressive cooling and drying of the global climate during the Cenozoic era. The relict populations of chameleons in southern Europe and the Middle East, including the common chameleon Chameleon that ranges from the Iberian Peninsula across the Mediterranean coast, are the diminished remnants of a formerly broader European distribution that once extended much further north. The extreme morphological diversity within the chameleon family is one of its most striking features. Chameleons range in size from the tiny Bruxia micra of Madagascar, which at around 29 millimeters in total length is one of the smallest reptiles known to science, to the Parsons chameleon of Madagascar, which can reach 68 centimeters and is among the largest chameleons in the world. Between these extremes lies an extraordinary range of body shapes, ornamentation styles, horn arrangements, casque forms, and color capabilities. This diversity has evolved relatively rapidly in evolutionary terms, with the Madagascar radiation in particular representing a spectacular adaptive diversification that has produced dozens of species occupying distinct microhabitats and ecological roles across the island's diverse forest types. The deep history of the chameleon lineage gives a useful perspective on what the animal represents today. The visual system, the color change mechanism, the ballistic tongue, the grasping feet, and prehensile tail. These features have been refined across tens of millions of years in changing forests on changing continents. None of them appeared fully formed. Each represents a trajectory of incremental modification from ancestral states, driven by selection pressures that we can only partially reconstruct from the patterns left in the genome and the fossil record. The chameleon we encounter today in the forest understory, sitting motionless in a patch of light, watching two worlds at once, is the current endpoint of a lineage that has been finding its way through forests for longer than most of what we call the modern world has existed. It wears its extraordinary features as naturally as the light it sits in. The chameleon's hunting strategy could be described, without oversimplification, as the art of not being noticed while noticing everything. It is a strategy built around a fundamental asymmetry. The chameleon, when still, is effectively invisible to the motion-detecting visual systems of most of its prey, while the chameleon's own visual system, continuously scanning through independently moving eyes, misses almost nothing that moves within its panoramic field. This asymmetry means that the longer the chameleon remains still, the better its information about the location and movement of potential prey becomes, while its prey's knowledge of the chameleon's presence remains near zero. Time spent motionless is not wasted time. It is productive time, accumulating information, and it costs relatively little energy from an ectotherm whose metabolic rate is modest at rest. This patience-based foraging strategy is sometimes called sit-and-wait predation, and it contrasts with the active search strategy used by many other predators that patrol their habitat looking for prey. Both strategies have costs and benefits that depend on prey density, habitat structure, and the predator's own energy budget. For an ectotherm in a forested environment where insect prey is patchily distributed but locally abundant when present, the sit-and-wait approach allows the predator to spend most of its energy budget on the tongue strike and digestion rather than on locomotion, keeping the overall metabolic demand very low. A single chameleon in a productive forest patch may make several successful strikes per day without traveling more than a few meters from its starting position, consuming what it needs with minimal energetic investment in finding it. The prey that chameleons most commonly take includes a wide range of insects and other arthropods, and in larger species, small vertebrates, including other lizards and occasionally small birds or rodents that come within tongue range. The selection of which prey items to target and which to ignore is not random. Researchers studying chameleon prey selection have found evidence for active discrimination based on prey size, movement pattern, and distance, with a tendency to target prey that falls within an optimal size range relative to the predator's own body size, and that moves in ways consistent with live prey rather than inedible debris. Very small prey may be ignored as energetically insufficient. Very large prey may be avoided as difficult or dangerous to subdue. The targeting process, mediated entirely through the visual system and executed through the tongue, represents a sophisticated real-time assessment of cost-benefit ratios. The integration of patience with precision is perhaps nowhere more apparent than in the full behavioral sequence of a successful chameleon strike, observed from beginning to end. It begins with the animal detecting a potential prey item with one eye, triggering the eye convergence response and a subtle shift in body orientation that may take 30 seconds or more as the chameleon repositions incrementally to face the prey. Then comes a period of binocular tracking, which may last minutes if the prey is moving unpredictably, with the chameleon waiting for a moment when the prey is stationary and at an optimal distance. When that moment arrives, the launch is instantaneous. But everything before it is patience, refined by millions of years of selection into something that looks almost meditative from the outside. There is an ecological consequence to the chameleon's patience strategy that extends beyond individual predation events. Because chameleons hunt without active searching, they do not disturb the vegetation around them or generate the kinds of movement that alert prey animals across a wide area. Their predatory footprint, ecologically speaking, is quiet. They take individual prey items without creating disturbances that flush other prey or trigger community-wide alarm. In this sense, their patience is not merely advantageous for themselves, but is structurally compatible with a certain kind of ecosystem dynamics, a soft, continuous removal of individual prey items from the available pool without the disruptive pulses that active predators create. The forest barely feels the chameleon passing through it, because the chameleon barely passes at all. The patience of the chameleon has, over the course of this animal's long evolutionary history, shaped everything about its body. The eyes are where they are because stillness makes panoramic vision more valuable than the directional vision needed by active searchers. The tongue is what it is because a sit-and-wait predator needs its strike to succeed on the first attempt, having invested so much time and thermal energy in reaching the moment of the strike. The color-changing skin exists partly because a motionless animal that can match its background is more invisible than one that cannot. The entire chameleon is, in some sense, a monument to the strategic value of doing very little, very well, for a very long time. Patience is not what the chameleon does while waiting to act. It is what the chameleon is. Chameleon reproduction involves one of the more unusual egg strategies in the lizard world. Most chameleon species are oviparous, meaning they lay eggs rather than giving birth to live young, though a significant minority of species, particularly those found at high altitude or in the cooler montane habitats of East Africa, are viviparous, retaining the eggs internally and giving birth to fully developed young. Egg-laying chameleons typically descend from their arboreal habitat to the ground for the brief an evidently uncomfortable process of oviposition. Digging a tunnel in soft soil or leaf litter, depositing a clutch of eggs at the tunnel's end, carefully backfilling the entrance, and returning to the trees as quickly as possible. The time spent on the ground is among the most vulnerable in a chameleon's life, and the behavior reflects this. The descent is fast, the digging efficient, and the retreat complete. The eggs themselves are leathery rather than hard-shelled, and they absorb water from the surrounding soil during incubation, increasing slightly in weight and volume as development proceeds. The incubation period is among the longest known for any lizard, with some species requiring 12 to 18 months, and the giant Parsons chameleon of Madagascar known to incubate for 2 years or more before hatching. During this long underground period, the developing embryos pass through a metabolic resting state called diapause at certain developmental stages, effectively pausing their own development in response to temperature and humidity cues from the environment. This developmental pause is thought to allow the hatching to be timed to favorable seasonal conditions, even when eggs are laid under variable conditions, ensuring that hatchlings emerge when food and thermal resources are optimal. The moment of hatching is remarkable to observe. After their long underground incubation, the hatchlings emerge fully formed and immediately capable of independent life. A baby Parsons chameleon hatching after 2 years underground is a miniature version of the adult, complete with the grasping feet, independent eye movement, and color change capability of the parent. It has never eaten, never seen light, never used its visual system for anything, and yet within minutes of emerging from the soil, it can climb, grip, track moving objects with independent eyes, and within hours or days make its first thumb strike at prey. The compressed developmental program that produces this competence, running for months or years in a small leathery egg in the dark earth, is one of the more quietly extraordinary achievements of reptilian development. Parental care in chameleons is essentially absent after the egg-laying event. The female provides no guidance, no protection, and no nutrition to her offspring after burying the clutch. The hatchlings must immediately begin the full chameleon life, finding their own food, managing their own thermal regulation, and navigating the visual and social world of the species without instruction. This independence from the outset means that all of the behavioral capabilities the young chameleon needs for survival must be available from the moment of hatching, either genetically encoded or rapidly self-assembled from a minimal experiential scaffold during the first days of life. The result is an animal that is, from its first hours in the sunlight, already everything it will need to be. The viviparity found in some chameleon lineages appears to have evolved multiple times independently within the group, in each case in species occupying cooler, more variable thermal environments. This pattern makes ecological sense. In environments where ground temperatures are too low or too variable for reliable egg incubation, retaining eggs internally allows the female to use behavioral thermoregulation to maintain a warmer, more stable developmental environment for the embryos throughout gestation. The trade-off is the energetic cost of carrying the developing young and the reduced mobility during pregnancy. But in high-altitude or high-latitude environments where cold soil is the alternative, the trade-off appears to favor viviparity, and natural selection has independently arrived at the same solution in multiple chameleon lineages facing the same thermal challenge. The deep diversity of reproductive strategies across the chameleon family from short incubation species in warm lowland forests to the multi-year diapause of Parsons chameleon and the live births of montane species, reflects the broad range of environments that the chameleon lineage has colonized over its evolutionary history. Each strategy is a local solution to the problem of timing offspring emergence to favorable conditions while managing the thermal and energetic costs of reproduction in that particular habitat. The egg buried in warm forest soil, the embryo resting in diapause through a mountain winter, the live young curled inside a high-altitude female, all represent the same underlying reproductive logic, expressed through the specific options available in each environment. The chameleon's patience, it seems, begins before hatching. There is a quality to the chameleon's presence in its habitat that is, for lack of a better word, integral. The animal does not impose itself on the forest so much as it participates in it, inhabiting the small vertical world of a few branches with a kind of ecological lightness that leaves almost no trace. Its predation is quiet, its movement is minimal, its thermoregulation is behaviorally mediated rather than metabolically expensive. Its coloration either matches or communicates through the very light that fills the forest. It is an animal that has found a way to be large enough to be effective and small enough to be nearly invisible, present enough to function and absent enough to avoid notice. In the ecology of the forest, the chameleon occupies a niche that feels almost like it was designed for exactly this animal. A forest ecosystem that hosts chameleons is itself structured around the same qualities of patience and layered complexity that the chameleon embodies. The canopy filters light in ways that favor animals adapted to reading structure in complex, dappled illumination. The insect community on which chameleons depend is itself distributed in patches and layers, organized by the thermal and chemical gradients of the vegetation, rather than uniformly across the available space. The predators that threaten chameleons, primarily birds with high visual acuity and motion sensitivity, exert selection pressure that favors exactly the slowness and background matching that characterize the chameleon's life. Every feature of the chameleon reflects the forest it lives in, and every feature of the forest shapes the chameleon that lives there. They have been co-evolving for longer than we have been watching. The threats to chameleon populations in the modern world are real, and in some regions severe. Deforestation removes the habitat that the chameleon's entire biology is organized around, and the animal's slow, arboreal lifestyle gives it very limited capacity to adapt to open, disturbed landscapes. Many chameleon species have small geographic ranges, particularly on Madagascar, where decades of forest loss have fragmented and reduced the habitats of dozens of endemic species. The international trade in wild-caught chameleons for the pet trade has historically placed significant pressure on wild populations of popular species, though captive breeding programs have reduced but not eliminated this pressure. Climate change, by altering the thermal environment and potentially disrupting the phenological timing of prey emergence, adds another layer of uncertainty to the long-term trajectory of chameleon populations in affected regions. Conservation efforts for chameleons must grapple with the specific challenges posed by animals that are difficult to census, depend on intact forest structure, have low reproductive rates, and are sensitive to relatively subtle changes in thermal and vegetational conditions. Effective conservation requires not just the protection of broad forest areas, but the maintenance of the structural complexity, the layered vegetation, the thermal gradients, the insect community that makes those forests habitable for chameleons specifically. A chameleon cannot simply be moved to a different forest and expected to thrive. Its requirements are precise, accumulated across a long evolutionary history of adaptation to specific conditions, and meeting those requirements in a changing world is a challenge that conservation biologists are still working to address. And yet, in the forests where they remain, chameleons continue to do exactly what they have always done. They warm in the morning light. They watch two worlds at once. They wait, with a patience that has no equivalent in our experience of waiting, for the moment when the tongue will fire and the prey will vanish and the stillness will resume. They change their colors in frequencies we cannot fully see, and communicate in a language whose richness we are only beginning to decode. They grip their branches with five-toed hands that have never lost anything they chose to hold. And at night, they pale and settle into rest, wearing a different face in the dark, watched by nothing, meeting nothing, complete in the simplest and most absolute sense of the word. And now, the forest can go quiet around you. The branch the chameleon is resting on is barely swaying in the night air. Its eyes are closed, the small scaled turrets smooth and sealed, the skin settled into its pale nocturnal arrangement, neither display nor camouflage, simply what the skin becomes when the day's signals have faded and the body is left to itself. Somewhere in the dark above it, the canopy holds still. The insects have mostly fallen quiet. The forest breathes in its slow way, and the chameleon breathes with it. You have been, tonight, in two directions at once. You have seen a tongue move faster than thought, and a skin that changes color through the physics of crystal. You have visited an eye that sees ultraviolet, and a foot that grips without effort, and an egg that waits 2 years to hatch. You have spent time with an animal that has been finding its way through forests like this one for nearly 100 million years, patient in ways that our species barely has language for, present in ways we are still learning to see. Let the room settle now, the way the forest settles after dark. Everything still finding its position, everything slowly arriving at rest. The chameleon is already there. It has been still, in the truest sense, since long before tonight. You can be still now too.